And
Yes.
can you hear me?
Yes.
Okay, welcome to the FAANG Workshop. I'm very grateful to be here to share with you about our project. Enhancer promoter interaction maps provide insights into skeletal muscle relevant traits in pig genome. And why are we doing this? First thing first, in the last decade, researchers around the whole world have done many GWAS research, and we got lots of GWAS leading signals. But deciphering the roles of these non-coding variants is limited and non-trivial because we have two major problems. The first one is that we do not have the detailed annotation of the cis regulatory elements in the tissues. And the second problem is that these cis regulatory elements often interact with their target genes over long genomic distances. Fortunately, we now have high quality annotations of cis regulatory elements like these two papers. But we still need chromatin conformation technologies to help us to identify the exact interaction between elements and their target gene. Here we choose EL-HICHIP for us to achieve this goal because through the enriched strategy, we can get a high resolution and lower cost than Hi-C, which is better for farm animals. And here we also add GRID-seg to detect RNA-chromatin interaction because you know that RNA is involved in multiple processes of transcription regulation. And here is our overall strategy and design. We collected loin muscle from large white animation, and we performed all these epigenetic experiment and to explain the GWAS leading signals. And we collected 248 SNPs and 14 QTLs from literature, and they can be divided into gross, mid-quality, and carcass relevant. And here we perform the differential loop regulated, differential chromatin regulated genes. As you can see that in large white, upregulated genes are mainly enriched in muscle development goal terms, while Meishan upregulated genes are mainly enriched in energy metabolism, which is consistent with our reality. And here we also predicted the transcription factor, and we can see that this result is the same. And we also performed artificial selection, and we can find that the aforementioned differential loop regulated genes are enriched in its own selective sweep regions, but not in the other breed. And we performed the integrated analysis for DNA-DNA interaction and RNA-DNA interaction. We can see that they have a similar trend and interaction levels. All these results can reveal breed specificity in finely tuned transcription networks, and this may provide epigenetic interpretation for genomic variants as well as GWAS leading signals. So here, we developed this step-by-step strategy to filter the candidate causal variants, and finally, for 15 muscle-related traits, we get 54 target genes and 223 candidate causal variants. And here are two examples. One is for the famous PRKG3. We identified a new candidate functional variant in the active promoter. And the second example is for the breeding value of FCR, and we identified a new target gene PRRX1 with two new candidate causal variants. And here is also another example of validation in cohort. This is for loin muscle depth, and we identified a new variation. And this variation, as you can see, that different allele have different effects in the herd. And here is our summary. We identified new target genes or genes that have a precise causal candidate causal SNPs. And the second is that around 75 enhancers will skip at least one gene to reach their target gene. This means that we need a chromatin conformation technology to help us to identify the exact target gene of GWAS leading signals. And the third one, we hope that we can provide new resource and new strategies for the pig community and for further studies. And here is our acknowledgement. We come from the Central China Agricultural University, and thanks and hoping to working with you together. Thank you. That's all.
Hey, great. Thanks very much.